The next speaker is Professor Sarah Rowland-Jones. She is a professor in the Oxford University. She has been a long time studying in the HIV-2 and Cd4 T-cell. Today he talks the HIV-specific Cd4 T-cell label. Please.
So I'd definitely like to thank the organisers for the invitation to this meeting, which I think has been really great. I think I want to thank them for the title that they've given me. It's taken some interpretation to work on this, exactly what to put. But I've been asked to give a historical context to the history of, to the story of HIV-specific CD8 T-cells. So I will mostly be talking about the work of other people, many of whom are in this room. And if you're in this room and I'm not talking about your work, please accept my apologies. It's a very big literature. So first of all, no conflict of interest to declare, and then to explain a bit about cytotoxic T-cells for the uninitiated. The reason that they play an important part in antiviral immunity is that they're able to recognize processed proteins from inside an infected cell. And because most of the proteins in a virus or pathogen are sampled and presented on the surface of T-cells, it means that the T-cells can then recognize the more conserved internal proteins, which is presumably why GAG-specific CTL have often come as a correlate of protection in HIV infection. And then having engaged with this exquisitely sensitive T-cell receptor on the HLA molecule holding these small peptides, a number of effector functions are triggered, and the relative importance of these differs in different infections, at least in murine models. But cytokines are produced, chemokines are produced, and the cell is also killed. So going back to the historical context, it's slightly appalling to see how long ago these papers were published, and indeed by some of the people on the stage. But the very first descriptions go back to the late 80s, and it was a time when you could simply describe the presence of T-cells in a condition and get a Nature paper, which was quite something. But you will notice many of the great and good names on this slide. And what I'd like to summarize on this slide is a lot of information accumulated over a long period that suggests that these cells are important in HIV infection. The virus is under pressure to drive the emergence of escape mutations. Seth Borrow and Rick Kaup showed quite early on the coincidence of these, the appearance of these cells with the rapid fall in plasma viral load following acute HIV infection. We and several other groups looked at the presence of these cells in highly exposed sex workers, which appeared to be enriched in the cervix in a cohort in Nairobi. And Grace John Stewart showed that the magnitude of this T-cell response in uninfected exposed infants correlated with protection from late breast milk infection. Then when tetramers came as a method of identifying T-cells, Graham Ogg found that there was an inverse correlation between the presence, the numbers of T-cells, and the plasma viral load. Animal experiments again showed that depletion of the cells led to a rise in plasma viral load in both acute and chronic infection. And perhaps intriguingly, these T-cells didn't just liberate new virus, they were able to kill virus-infected cells before new virus is produced. And then, as Galit mentioned earlier, there's been a consistent relationship between HLA molecules and good outcome, and this was shown in the various GWAS studies, which you mentioned earlier. However, they're not all good, and several other studies looking at the overall response using gamma interferon secretion as a readout rather than tetramers showed simply no association between the magnitude or the breadth of the response and any clinical parameters. And it became clear that HIV has mechanisms to reduce the ability of T-cells to recognize infected cells, such as the ability of NEF to downregulate class 1 molecules. And the virus escape appeared to be something that was happening on a large scale, shown to happen within weeks of primary infection, and leading to population-level changes in the virus, which in a seminal paper was shown to undermine the protective HLO associations of B51 and good virus and good outcome with HIV in Japan over a period of time. And again, other people in this room have shown that these T-cells had an unusual differentiation state. They weren't particularly good at killing. And then increasingly it's become aware that they are expressing a number of inhibitory molecules that are associated with senescence and exhaustion. That's not what I wanted to do. However, again, studies of elite controllers have suggested that there are particular features of a good T-cell response, and in particular responses to the GAG protein, the levels of polyfunctionality, the proliferation of these T-cells, and the avidity of the T-cells. That's ability to recognize target cells at very low antigen concentration. And in particular, what seemed to prove to be quite a useful readout in a number of situations, increasingly in vaccine studies, is the ability of T cells to suppress viral replication in vitro assays. And this may be related to selection of particular T cell receptors. But then we, I think I was... Then we went through the bleak years of T-cells that followed the STEP trial, which at the time was thought to be the best and most immunogenic T-cell inducing vaccine. And as you probably all know, the trial was halted because interim analysis showed no protection and possibly even an increased risk under certain circumstances. And although at the time these T-cell responses were thought to be quite respectable, we would now regard these as really very modest T-cell responses with very limited breadth. And for a while, the whole focus shifted away from T-cells and towards antibodies, and that's something that I think many of us can relate to. However, there was a potential, these important studies of very early events in macaque infection have suggested, from Ashley Haas's group, have suggested that there is that tiny window of opportunity following mucosal exposure when there are just small foci of infection in the mucosa, many of which can be extinguished by the influx of T cells if the T-cell response is large enough, suggesting that if you had a presence of T-cells at the site of mucosal exposure, you might be able to contain and eliminate infection. So now we come to the reborn part of T-cells, and I want to highlight studies, several of which have been presented at this meeting. And in particular, the much more detailed studies now of the very early events of acute infection. One of the first of these comes from the FRESH cohort in Durban, where women were identified before the peak of viral load in the very early stages, and at this particular protocol, they weren't given antiretroviral treatment. And the data quite clearly show that there is an inverse correlation between the frequency of activated T cells, which in this study were largely HIV specific, and the set point viral load. However, they did note that these T cells had relatively poor at producing interferon and highly susceptible to apoptosis. And then presented very nicely yesterday by Liddy Troutman in studies from Thailand in the SEARCH cohort, again showed in patients who were treated very early, again showed very high-level T-cell responses that, but not appearing until about big stage 3, and again showing an inverse correlation here between the level of activated T-cells and the plasma viral load fall in response to antiretroviral therapy, with an inverse correlation with the reservoir size. So even in the presence of treatment, these T cells were developing and exerting some control. But there's still a suggestion that these responses are coming in too late in natural infection to make the difference. In vaccines, again, we've heard about from this meeting, particularly from Dan Brook and Tom Hankey's groups, that these newer generations of vaccines, far advanced from the original Merck vaccines, with priming with particular strains of adenovirus and boosting, perhaps also with protein boosting to produce antibodies, are able to elicit much stronger, much broader responses that in this panel here you can see inhibit viral replication in vitro of several different clades. And the insertion of predominantly conserved parts of the virus into vaccines has allowed the refocusing of T-cell responses towards very conserved regions of HIV, which are presumably resistant to escape mutation. And as Galit showed earlier, certainly in Dan's studies in macaques, they have provided significant protection against virulent challenge. And even more intriguingly, we're raising lots of interesting scientific questions, has been the work of Louis Picker and Scott Hansen, looking at a CMV, attenuated CMV vector that's adapted to replicate in fibroblasts, leads to clearance in around half of or over half of the vaccinated macaques. And these CD8 T-cells use unusual restriction elements, particularly HLA-E and class 2, and this is something that exactly how this happens depends very much on the CMV genes that have been removed from the vector, and it's still not quite clear whether this can be reproduced yet in humans. But again, I think T-cells have been reborn in the cure strategies. It's clear that killing reactivated virus that's been awoken by latency reversing agents is likely to require HIV-specific CTL, and particularly those with the characteristics of elite controller. And we heard this morning about checkpoint inhibitors such as PD-1, anti-PD-1, that may allow these T-cells to fulfill their potential in vivo. So finally, I just want to finish with some work from our group that suggests that T-cells that we see in HIV-2 infection may have particular characteristics that would be able to associate with protection. And in HIV-2, as you may know, there's a much higher proportion of the elite controllers, which has a clinically stable phenotype, able to maintain undetectable viral load for at least a decade. And these people are very clearly distinguished from the progressors by the magnitude and the specificity of their T-cell response, whereas CD8 T-cells targeting GAG are often absent in progressors. Working in collaboration with Victor Appé and Assier Sirion last year, we were able also to show similar results in a French cohort, and in particular Assier's assay to look at the viral suppression of viral replication showed particularly potent replication of inhibition of viral replication from cells from the HIV-2 patients. And there were other striking features that suggest preserved relative preservation of the immune response in these patients. So why do I think HIV-2 specific T-cells are worth examining further? They are at an earlier stage of differentiation with preserved ability to proliferate. They're able to persist at very high frequencies in patients with undetectable plasma viral load, and it's not really clear how these are maintained. They work at extraordinarily high functional avidity, 1 or 2 logs higher than we see with most other antiviral specific T-cells, and they show potent suppression of HIV-2 replication in vitro. So to summarise then, I think T-cells do play an important role in the control of HIV replication in vivo, and we've seen that particularly now in these lovely studies of the early events of acute HIV infection. And they clearly are going to be part of vaccine strategies and potentially of cure strategies. But what we need to do is improve on simply generating high numbers of T-cells, but also eliciting T-cells of real quality that give us the features that we want. And I would argue that part of this is seen in the T-cells that we see in naturally infected people with viral control in HIV-2 infection. So thank you very much for your attention. Stop there.
Thank you very much. Unfortunately, we don't have enough time to discuss. Probably the last section that we discussed. If you have any questions for Sarah. Sorry.
Thanks, Sarah. So next speaker is Lisa Chakrabarty, who is Director de Recherche, Associate Professor here at the Pasteur Institute in Paris. Lisa.
So good afternoon, and I do thank the organizers for this opportunity. Today I'll discuss how TCR clonotypic analysis could be used to study HIV-specific CD4 responses and the implication for vaccine design. But before, the organizers asked me to discuss the double phase of CD4 T-cells, because as you know, they are key immune effectors, but they are also preferred HIV targets. And this poses a dilemma when one wants to induce antiviral CD4 responses. So first, a brief reminder of the central role of CD4 T-cells in adaptive responses. CD4 T-cells provide help to multiple immune effectors, including B cells, dendritic cells, which then become licensed to help CD8 T-cells, also innate cells. A function that is often neglected is that CD4 T-cells attract other immune effectors to infection sites because they produce abundant chemokines. And the issue is that when we are in a situation of a lack of CD4 T-cell help, as you well know, basically there is progression to immunodeficiency and AIDS. If we focus on the fate of CD8 T-cells in the absence of CD4 T-cell help, one can see that these cells show multiple signs of immune exhaustion, including decreased proliferation, loss of polyfunctional cytokine secretion, upregulation of inhibitory receptors, increased susceptibility to apoptosis. And these helpless CD8 T-cells also show a propensity to kill each other through fratricide because they upregulate TREL. So evidence from multiple models indicate that CD4 T-cell help is needed to sustain CD8 memory responses in the setting of chronic viral infections. So the issue, obviously, is that activated CD4 T-cells are preferred HIV targets, and in particular, that HIV-specific cells are more prone to, even more susceptible to HIV infection and depletion. And this may be because dendritic cells, which captures HIV and presents HIV antigens through MHC class II, has seen the virus and is thus more likely to be infected. So upon formation of the immunological synapse with an HIV-specific CD4 T-cells, the virus is more likely to be transmitted. And then, upon activation through TCR signaling, there will be massive replication of the virus and depletion of the very cells that are supposed to orchestrate the anti-HIV immune response. There is evidence that CD4 T-cell responses are impaired very early on in primary HIV infection. So if one treats HIV-infected patients very early, CD4 proliferative responses can be maintained. However, upon treatment interruption, in most cases, the virus will rebound, and there's no correlation between the CD4 proliferative responses and the delay to rebound. Another issue is that HIV-specific CD4 T-cells may even facilitate HIV acquisition in certain cases upon vaccination. This has been shown, for instance, by the group of Guido Silvestri in the simian model, where they could show that the frequency of activated CD4 T-cells that are CCR5 positive and that are present in the mucosa upon vaccination does correlate with the risk of SIV acquisition. And there were also signals in the therapeutic vaccination trial with Alvac that induction of CD4 responses could be deleterious. We actually don't really know how to induce antiviral CD4 responses that would not be associated with this risk of fueling viral infection. But I think we may learn from the HIV controllers, also called elite controllers, because their CD4 T-cells are qualitatively different. They do have preserved central memory responses associated with high proliferative capacity, high IL-2 secretion, low FOXO3A expression, and lack of immune exhaustion markers. But this is not unique to the controllers, because patients treated early in primary HIV infection do have these responses. And also, patients treated in the long term do recover proliferative CD4 responses. So what is more unique to the controller group is the persistence of effector memory CD4 responses in spite of the very low antigen amounts that are present to induce these effector responses. So this translates into polyfunctionality, the presence of highly differentiated Th1 effectors that can degranulate, and it has been shown by the group of Hendrik Streck that the presence of CD4 T-cells with cytotoxic potential in the primary infection phase correlates with a lower viral load in the chronic phase of infection. Another peculiarity of controller CD4 T-cells is that they may be less infectable, as shown by the group of Asier Saïs-Sirion and also by that of Matthias Lichterfeld. I should say, however, that this is debated by other groups, but one may speculate that maybe the Th1 differentiation status, which involves interferon gamma production, may lead to ISG induction, and that could make these cells less susceptible. So we think that a key reason for the particular properties of HIV controller-specific CD4 T-cells is that they have high TCR avidity. So to do this study, we analyzed the responses to the most immunodominant CD4 epitope that we call GAG293. We amplified primary CD4 cell lines specific for GAG293, and then we labeled the specific CD4 T-cells with MHC tetramers, MHC class 2 tetramers. And we could do tetramer dilution assays, and we observed that controller CD4 T-cells could bind the tetramers more efficiently than those from treated patients, as indicated by a lower last positive tetramer dilution. So this means that the TCRs were intrinsically different in the controller CD4 T-cells, and that these cells had the capacity to respond to minimal amounts of GAG antigens. And this may well explain why Th1 effector persists, because they will be sensitive enough to detect this very low amount of GAG antigen. And this also has implications for immune reconstitution, because it seems that treated patients have lost these high-quality CD4 T-cells. And if one aims at reconstituting the CD4 T-cell pool, one needs to think not only of the quantity of CD4 T-cells, but also of their quality. So because the TCRs appear different in HIV-specific CD4 T-cells of controllers, we set to characterize them at the molecular level. So we sorted the GAG293 tetramer-positive cells, and we analyzed their TCR repertoire. We found that particular TCR variable genes, TRAF24 and TRBV2, were preferentially expressed in the GAG293 specific cells, and more so in the controller group. We then sequenced the CDR3 region, which is these hypervariable regions that makes the most contact with the peptide MHC complex here. And what we found was that many of the CD3 sequences, which we called clonotypes, were shared between patients. And they were these shared TCR sequences, called public clonotypes, were more prevalent in the controller group than in that of treated patients. We could define motifs that were more prevalent in the clonotypes of controllers, and we could also quantify the public clonotypes and show that they were more abundant, both for the TRAF24 and the TRBV2 chains in the controller group as compared to the treated patient group. So this meant that public clonotypes could play a role in the high avidity recognition of GAG293. So to test this notion, we tested the function of the most prevalent GAG293 specific public clonotypes. But before that, I wanted to show a few results from the single-cell analysis, where we sorted the tetramer-positive cells directly ex vivo and analyzed them by multiplex real-time PCRs with the Fluidigm technology. And you can see here the results for about 600 single cells, and we looked at TRBV2 expression, and it's quite visible that TRBV2 is more prevalent in the tetramer-positive cells than in the tetramer-negative cells. And furthermore, that TRBV2 within the tetramer-positive cells is more prevalent in the controller group than in the treated patient group. If one looks at TRAF24 co-expression, one sees that most of the TRAF24 positive cells are also TRBV2, though the reverse is not true. Some of the TRBV2 positive tetramer cells express other TRAF genes. So these analyses reassured us that we had sorted the right tetramer-positive cells, even though they can be quite rare, and that it confirmed that the TCR bias was very marked in the tetramer-positive cells. So then, we tested the most prevalent public clonotypes from controllers functionally by inserting them in a bicistronic lentiviral vector. And I will go rather briefly on these results because they were published last year. But what we found was that these highly prevalent GAG293 specific public TCRs could confer broad HLA cross-restriction, meaning that a single of these TCR could recognize the GAG293 peptide in the context of multiple HLA-DR alleles, up to 5 DR alleles. And this could explain why they could be shared by HIV controllers of diverse genetic backgrounds. These TCR also had high TCR affinity, and this was shown by our collaborators at Monash University, Stephanie Gras and Jamie Ross-Jones, who could express the soluble TCRs and measure their affinity by SPR. And they found affinities that could go below 1 micromolar, which is in the highest range known for class 2 restricted TCRs. These high-affinity TCRs conferred high antigen sensitivity. There was a good correlation between the two parameters, and they also conferred polyfunctionality, as indicated by secretion of TNF, MIP1 beta, IL-2, interferon gamma, and they also had the degranulation marker CD107A. So the simple transfer of a high-affinity public TCR was sufficient to recapitulate the functional properties characteristic of HIV controller CD4 T-cells. And more recent data support this notion. This study of the cytotoxic capacity of the TCR transduced cells was done by Moran Galperin. What he did is infect monocyte-derived dendritic cells with a single-cycle HIV GFP reporter virus, and then incubated these DC with TCR transduced CD4 T-cells that were autologous. And when you put these two cell types in contact, you see that the number of infected DC decreases here in the presence of the high-avidity F24 TCR as compared to the mock transduction. This effect was effector-to-target ratio dependent, and also it depended on the affinity of the TCR, as shown here by comparing 3 TCRs of decreasing affinity. So this finding suggests that the high-affinity CD4 T-cells may directly contribute to HIV control by eliminating MHC class II positive HIV-infected cells. And what was interesting was that when we transduced the TCRs in CD8 T cells, we could also see cytotoxicity, but in that case, the affinity threshold was higher, and only the very high affinity TCR did confer this property. So what we think is going on is that in a regular CD4 T cells, the CD4 co-receptor can bind MHC class II and bring a pool of LCK molecules which will promote TCR signaling. But in a CD8 context, the TCR is still MHC class II restricted, but CD8 co-receptor cannot bind MHC class II, so the pool of LCK is not brought to the TCR. And only the very high affinity TCR with sufficient signaling capacity by themselves can trigger cytotoxicity. So we think these very high affinity TCRs may be interesting for immunotherapeutic applications because their transfer can confer cytotoxicity to both CD4 and CD8 T cells. And this is a notion we want to test in a humanized mouse model. So how do we apply these findings to the field of vaccinology? We asked whether, yes?
You're giving a really interesting talk, but it's gone a long time over,
Okay.
so perhaps you could wrap up.
So I will wrap up by saying very briefly that in the ADVAX vaccination trial, we analyzed a few of the volunteers, and we could find many of the public clonotypes shared between the vaccinees and the controllers, meaning that it will be feasible to compare the frequency of shared clonotype between the repertoires of vaccinee and controllers. And since we showed that these public clonotypes can confer interesting properties, this may be informative on the quality of the CD4 response. And I will just mention these two papers recently published in Nature by Mark Davis groups and Paul Thomas group, saying that it becomes feasible to predict antigenic specificity by analyzing the sequences of the TCR repertoire. So now we may have a new tool that may help predict vaccination outcome by analyzing the TCR at a very early stage in HIV vaccination trials. And I will acknowledge the people involved in this work, in particular Moran Galperin, Daniela Benatti, Madhura Mukhopadhyay, and Mathieu Clairaut, as well as Olivier Lambot, the PI of the HIV controller cohort, and all of our other collaborators and the patients. Thank you for your attention.
No question. Sorry that we don't have enough time for it to be discussed in the later section. But okay, next speaker is Professor David Evans. He is a professor at University of Wisconsin-Madison. So he talked He talks mechanisms of the HIV-1 resistance to the ADCC. Please.
Okay, thank you. So we have no conflicts. So as an introduction, I'll start off with a couple of basic principles that reflect our perspective coming into this field on the role of non-neutralizing functions of antibodies in the ability to target HIV-infected cells, and particularly the role of antibodies in killing virus-infected cells by antibody-dependent cell-mediated cytotoxicity. The first is, of course, HIV has evolved to replicate continuously in the presence of ongoing and active host immune responses. And therefore, by definition, has acquired mechanisms to overcome all immune responses that are made in most infected individuals, and this includes ADCC. The second is, if we've learned anything from the neutralizing antibody field over the past 20 years, that is, the ability of antibodies to block HIV infectivity is critically dependent on their capacity to bind native functional envelope trimers as they exist on virions. And if we're talking about protection by neutralizing antibodies, this means envelope on the surface of primary isolates in transmitted founder viruses. So when we started in this area, we set out to develop an assay that would allow us to measure the ability of antibodies to kill HIV-infected cells expressing the native trimeric form of the viral envelope. And the assay that we developed is depicted here. This was work that was done a few years ago by a former graduate student in my laboratory, Michael Alpert. It's based on a pair of stable cell lines. The first is a CD4 positive T cell line that contains a TAT-inducible luciferase reporter gene. So when these cells become infected with HIV, the viral TAT protein turns on luciferase and provides a readout of infectivity. If we incubate these cells together with a natural killer cell line that constitutively expresses CD16 in the presence of serial dilutions of antibody or plasma, we can measure ADCC as the dose-dependent loss of luciferase after an 8-hour incubation. And so most of the data that I will present during this presentation was generated using this assay. And work by our laboratory and a number of other groups have identified several molecular mechanisms that help to explain how HIV-infected cells avoid elimination by antibodies in chronically infected individuals. The first is that there's actually not a lot of viral envelope glycoprotein on the surface of an infected cell. This is an immunofluorescent image of HIV-infected cells that were permeabilized and stained for the viral envelope glycoprotein. You see that most of the envelope staining is occurring in perinuclear regions corresponding to the ER and Golgi. There's some punctate staining in endosomal vesicles, but relatively little on the surface of the infected cell. This reflects the effects of cellular trafficking motifs in the cytoplasmic domain of envelope, in part. And one of the best characterized of these is a tyrosine-based endocytosis motif close to the transmembrane region of the protein. It's been known for quite some time that if you introduce mutations into this conserved AP2 binding site, the result will be an increase in envelope expression on the surface of infected cells. And we showed, not surprisingly, that these mutations also increase the susceptibility of infected cells to ADCC. So in this assay, ADCC is measured in percent relative light units on the y-axis. Antibody concentration is from high to low, as shown on the x. The dotted line indicates a 50% reduction in luciferase, or 50% ADCC. So the point at which these curves cross the dotted line reflects an antibody concentration for half maximal killing. So this assay is showing us that if you make glycine or phenylalanine changes into this endocytosin motif, it increases the sensitivity to killing by well over a log. Our group also works on another restriction factor for HIV known as tetherin. This was a factor first identified in Paul Binash's group. Tetherin is an interferon-inducible transmembrane protein that has a rather peculiar topology that allows it to, it has an N-terminal and a C-terminal membrane anchor. And when it's expressed on the surface of infected cells, it can become incorporated into budding variants and prevent their detachment. Well, HIV normally overcomes restriction by tetherin by expressing VPU. And whereas tetherin is a type 2 transmembrane protein, VPU is a type 1 transmembrane protein, physically interacts with the membrane-spanning domain of tetherin and mediates the down modulation and degradation of tetherin, simply removing this factor from sites of assembly and virus release. Well, in the absence of VPU, viruses become captured by tetherin and accumulate on the cell surface. And it occurred to us that this might make the cells susceptible to recognition by antibodies. And so we simply asked, how does the susceptibility to ADCC compare for cells infected with wild-type HIV versus a VPU-deleted strain? In this assay, we found that deletion of VPU increases the sensitivity by approximately 50-fold, indicating that, in fact, VPU is protecting HIV-infected cells from ADCC. But it doesn't tell us why. Like other proteins of the virus, VPU has multiple functional activities. In addition to down-modulating and degrading tetherin, it can also down-modulate Cd4 and NTBA, so Cd4 being, of course, the primary receptor for viral entry, and NTBA is a stimulatory molecule required for NK cell recognition. So in principle, any of these activities could contribute to the resistance of infected cells to ADCC. Well, we demonstrated by RNAi knockdown that knockdown of tetherin in cells infected with wild-type and VPU-deleted virus increased resistance to killing, whereas RNAi knockdown of CD4 and NTBA didn't have an effect. So this suggests that it's the anti-tetherin activity of VPU that's preventing the accumulation of captured particles on the surface of the infected cell that's contributing to resistance to antibodies. Another mechanism was identified by Andreas Finzi's group and others, showing that the down modulation of CD4 by VPU and NAF contributes to resistance to recognition by antibodies. So CD4 down modulation is a well-known overlapping function of these two viral proteins. And in cells infected with NAF or VPU-deleted virus, or particularly when both of these gene products are absent, CD4 is retained on the cell surface, and it can engage the envelope glycoprotein and trigger a conformational change that exposes CD4-inducible epitopes that can then be targeted by antibodies that are abundant in the plasma of most infected individuals that can then mediate the killing of these virus-infected cells. So the prototypical member of this class of antibodies is a monoclonal antibody known as A32. A32 binds to a CD4-inducible epitope on the inner domain of GP120 that's normally occluded in the closed, unliganded form of the trimer. When we tested this antibody for recognition of a transmitted founder virus, the wild-type virus was quite resistant to ADCC. But in cells infected with the same virus containing deletions in both VPU and NAF, we see a dramatic increase in sensitivity to killing by this antibody. Well, these types of antibodies are quite abundant in plasma. When we compare the ADCC activity of plasma from 10 HIV-positive donors for the ability to kill the wild-type transmitted founder virus, we see very little activity. Whereas the cells infected with the NAF and VPU-deleted virus are very sensitive to killing. So again, this is reflecting, in the absence of these two accessory proteins, the inability to down-modulate CD4, which is when present on the cell surface, can induce this conformational change in envelope and expose these epitopes that are otherwise occluded in the native trimer. Well, in addition to these mechanisms, there are a number of structural features that help to explain the resistance of HIV to antibodies. The trimer, outer domain surfaces of the trimer are covered by extensive end-link glycosylation that form this glycan shield. It makes it very difficult for most antibodies to penetrate and bind to the underlying polypeptide. Surfaces of envelope that must be conserved for functional reasons to mediate entry, the CD4 binding site and the co-receptor binding site, are concealed by conformational mechanisms. So the CD4 binding site is a deeply recessed pocket that is inaccessible to most antibodies, and the co-receptor binding site isn't even formed prior to CD4 engagement. So these features help to explain why it's so difficult to elicit an antibody response to the native trimer that's able to mediate the neutralization of genetically diverse primary isolates. Well, there's been a revolution in recent years in the isolation of roughly a couple dozen, depending on how you count them, potent broadly neutralizing antibodies that are able to recognize conserved conformational features of the trimer and neutralize a majority of primary isolates. So we tested these antibodies for the ability to kill HIV-infected cells in our assay. These included antibodies to the CD4 binding site, proteoglycan epitopes in the V2 and V3 regions, and a proteoglycan epitope in the at the junction of GP120 and GP41. And we included cells infected with lab-adapted NL4-3, so this is a virus that has lost many of the structural features that confer resistance to neutralization during passage and culture. Cells infected with HIV-GRFL, which is a tier 2 neutralization-resistant primary isolate, typical of most naturally transmitted viruses, and SIV-MAC239, which is we've included as a negative control. And consistent with the potent, broadly neutralizing activity of these antibodies, they were all able to mediate the killing of cells infected with our primary isolate GRFL. And in most cases, we're quite good at killing NL4-3 infected cells. But when we did the same experiment, this time with a panel of non-neutralizing antibodies, including A32 and C11, which again recognize CD4 inducible epitopes on the inner domain of GP120, B6, which is a non-neutralizing epitope in the CD4 binding pocket, 17B and X5 target the CD4 inducible epitope in the co-receptor binding site, and these antibodies to immunodominant epitopes in GP41, they're normally occluded in the closed trimer. Whereas several of these antibodies were able to efficiently kill cells infected with a lab-adapted virus, none of them exhibited any measurable activity against cells infected with JRFL. So in fact, ADCC correlates quite well with neutralization. This correlation is strongest for cells infected with JRFL, or JRFL in general, because in this case, all of the antibodies that were able to mediate the killing of cells infected with this virus also neutralized JRFL. So in conclusion, there is very little envelope on the surface of HIV-infected cells, and this is due in part to trafficking motifs in the cytoplasmic domain of GP41 that carefully regulate the amount of envelope present on the surface of the cell prior to the assembly and release of infectious virus particles. Tethering down modulation by VPU prevents tethering from capturing particles at the cell surface that would otherwise render the cell susceptible to ADCC. Cd4 down modulation by both NEF and VPU remove the primary receptor for the virus that when it's present on the cell surface can induce a conformational change and expose epitopes and envelope for recognition by antibodies that are quite common in plasma. And the correlation between ADCC and neutralization suggests that many of the same structural features that confer the structural features of envelope that confer resistance to neutralization also confer resistance to ADCC. So these are together, I think these features help to explain why HIV-infected cells are quite resistant to antibody-mediated killing. And these are mechanisms of resistance that we're going to need to consider and overcome in the development of vaccine approaches to optimize antibody-mediated responses against HIV-infected cells. So I'll end there and thank those who have contributed to this work. Thank you.
Thank you very much. We will discuss in the last part, right? Thank you.
Okay.